Welcome to this episode of That's Good Science, where we showcase scientists who explore the possibilities of gene expression analysis. Today, we investigate the surprising variability found among seemingly identical cells. Until recently, scientists who wanted to understand whole genome changes in transcription had little choice but to study a group of cells. They simply didn't have the tools to study individual cells. While many advances were made, important questions lingered. What if individual cells within a group respond differently to the same stimuli? What more could we learn by studying single cells? Alex Shalik, Rahul Satija, and their colleagues at the Broad Institute of MIT and Harvard tried to answer these questions using bone marrow-derived dendritic cells. BMDCs are an important part of the early immune response. They present antigens to activate B and T cells in the presence of a pathogen. Many studies have reported gene upregulation in BMDCs in response to pathogens, but it has been unclear if every BMDC turns on all of the upregulated genes, or if some of the cells turn on one subset of genes while other cells turn on another. Shalik and colleagues used the Smarter Ultra-Low RNA Kit for Illumina sequencing to see how the transcriptome in a single BMDC is affected after it's stimulated with a particular bacterial antigen, LPS. This kit allowed the researchers to perform RNA sequencing on single cells and, even better, to obtain full-length transcript data that identified differences in mRNA splicing among those cells. They found extensive differences between individual cells both at the level of gene expression and in splicing patterns, which wouldn't have been possible to identify with other single-cell RNA sequencing kits. This work reminds us that even seemingly well-understood transcriptional responses have hidden complexities waiting to be discovered. All that's needed is a closer look with the right tools. And that's good science.